How's it going, everyone? It's Chow here, and today we are going to talk a little bit about pyruvate oxidation. This is a relatively short section, so I'm not even going to bother with the webcam, so you don't get to see my face over here, unfortunately, but let's get started. So when it comes to pyruvate oxidation, it happens actually right after glycolysis. So if you think back to glycolysis, you have all these little steps that start off with a 6-carbon sugar, so glucose, and you break it down into two 3-carbon molecules called pyruvate. So you have pyruvate here as a 3-carbon molecule, and then another 3-carbon molecule, pyruvate. So you get two 3-carbon products called pyruvate. You lose 2 ATP during the process, during the entirety of the process, but you end up gaining 4 ATP at the result, as well as 2 NADH molecules. So you get 4 ATP molecules as well as 2 NADH molecules, but you use 2, so you end up with a net gain of 2 ATP. So there you go, just some steps of glycolysis, but afterwards we have to kind of process that pyruvate. Sometimes we call it the preparatory step, but it's a step that basically shifts and changes the pyruvate a little bit into something else before we can actually run the reaction. So what happens during pyruvate oxidation is that you have this particular enzyme complex called pyruvate dehydrogenase or pyruvate dehydrogenase complex, and you run that particular pyruvate molecule through this process. It's kind of a decarboxylation, so you end up losing carbon dioxide in the process, but you ultimately make this particular molecule here called acetyl-CoA. So it's basically an acetyl group that's attached to a CoA enzyme. So what's really interesting, of course, is that you have one glucose that makes 2 pyruvate, and each pyruvate makes 1 acetyl-CoA. So ultimately, you end up with 2 acetyl-CoA from 1 molecule of glucose. And this is basically what happens. You're turning a pyruvate molecule, which you get at the end of glycolysis, into acetyl coenzyme A or acetyl-CoA, and it's done by a complex called pyruvate dehydrogenase. So it's a multitude of different enzymes. So it's an enzyme and a multitude of different cofactors such as TPP or FAD, and it's also a redox reaction. So if you look at it, it's really interesting because you have this particular group over here, which kind of looks like a carbon dioxide, doesn't it? And what's really interesting is through this process with pyruvate dehydrogenase, you end up actually removing this OCO over here, which is indeed carbon dioxide. So what's really fascinating is that with this process, you start off with a pyruvate molecule over here, and then you make an acetyl-CoA, and you lose carbon dioxide in the process. What's also very interesting is that, let's not forget, this is a redox reaction. So we have to strip some electrons or add in some electrons somewhere. And this is done with NAD+, which ultimately, through the process, you create NADH. So NAD+ is actually reduced into NADH with this process. So ultimately, with pyruvate to acetyl coenzyme A, you make CO2, which is honestly a waste product, and then you also make NADH. In addition to that, the pyruvate is converted into acetyl-CoA. Now remember, you have 2 pyruvate molecules that result at the end of glycolysis. So that means you actually end up making 2 coenzyme A's, which means you actually end up making twice the amount of those products. So with each particular pyruvate, you make 1 CO2 and 1 NADH, but since you have 2 pyruvate from glycolysis, then you actually run that twice in a way, and you end up making basically 2 CO2 molecules and actually 2 NADH molecules. So basically, you're actually taking pyruvate again and turning it into acetyl coenzyme A, and then that acetyl coenzyme A will ultimately enter into the Krebs cycle, and that's where you can basically do more conversions, make more NADH, lose more CO2, and then ultimately use those NADH molecules in the electrons transport chain, which we will talk about in another lecture. So that's basically it. I hope you all enjoyed this. This shouldn't be too difficult. You're just turning pyruvate into acetyl-CoA and you're creating some CO2 and an NADH in the process, and it's mediated by a pyruvate dehydrogenase complex.